everyone. This is my lecture to Pharmacy third semester students entitled Introduction to Subterranean Organs, presented by me, Dr. Shema Abdelgawit, Lecturer of Pharmacognosy, Faculty of Pharmacy, Fayoum University. In this lecture, I will give you a brief hint or explanation of the what is the subterranean organ definition, subterranean organ types or classification and their functions, as well as their microscopical features and morphological description. As we previously studied in the last semester, the plant constituted of several organs. Some organs are aerial, such as leaves, flowers, and stem, while other organs below under the ground, such as the roots. So, simply we can define subterranean organs as the organs that grow under the ground or near the soil. There are several important functions for the subterranean organs, including the following. At the first, they are a great mean of vegetative propagation or sexual reproduction. As you know, there are two types for plant to propagate or reproduce, such as the sexual reproduction using the flower organ, using pollination between the pollen grains and the gynecium in other flowers, the female and male flowers. This is called sexual reproduction. And the other way is the asexual reproduction or vegetative propagation, which is done by the mean of using part of the root or the complete root. For example, as you know how we can cultivate potato. Nobody bought the seed of potato to cultivate this plant, but usually what happened that the farmers put one tuber of the potato under the soil and irrigate them with water, and after that the tuber forming buds which constitute another potato or another plant for what is known as budding. And potato, as we will see in the next slides, is one of the types of subterranean organs. Also, usually we did that, for example, when cultivating ginger, we add just a slice of the ginger rhizome under the soil, and then it will constitute a new plant, and so on. So, subterranean organs is very important in the vegetative propagation or sexual reproduction of the plants. The second important function of the subterranean organs is that they are very important to absorb water and soluble nutrients and vitamins, minerals from the soil, and transporting them to the stem, leaves, or the aerial parts of the plant. They have essential function for the support or anchoring the plant to the ground, because they are, when we will see their microscopical investigation or elements, we will see that it is full with the lignified elements such as xylem vessels, fibers, trapeze, and even sclerites, which have a great role in the support of plant in the ground, as well as their function, of course, to transport water. The last important function for the subterranean organs is that they serve as storage organs for the reserve of food materials as carbohydrate. As we know, the aerial parts such as leaf can synthesize the carbohydrate using the photosynthesis, and then this food material, for example the starch, is transferred to other organs of the plant through the phloem tissue. Usually, a plant forming high amount of reserve food material use the roots or the subterranean organ as the storage room or organs for this reserve food material. For example, we can see large amount of starch present in the rhizomes or roots of rhubarb and also in ginger, and also some sugars are stored, such happen in gentian root. Because of the vegetative propagation, absorbing water and the nutrients, supporting the plant, as well as storing the reserve food material, subterranean organs play an important role for the plant. Actually, subterranean root is not only the subterranean organs are not only the root as we commonly know. There are two main types for subterranean organs, named as subterranean stem and subterranean roots. And later on, we will discuss the subtypes of subterranean stems, such as rhizomes, corms, tubers, and bulbs. There are no differences between the subterranean stem and roots in commercial sense, but in pharmacognostical point of view, there are differences either in morphology or histology of the two organs, as we will discuss in the next slides. Now we will start discussing both morphology and histology of subterranean stems. As we told in the previous slide, we don't have only subterranean root, but another type called subterranean stem. So what make us define this part as stem, not root? Because there are a lot of similarities between this organ and the aerial stem. While it's these characteristics, as we will see later, not found on the normal root, so this organ classified as subterranean stem. What are the similarities? It's similar to the aerial stem. It's divided to nodes and internodes, as we can see here in this picture. It's divided to nodes and internodes, such as the aerial stem, and also there is a bud at each node. In the aerial stem, the bud forming the leaves or the flower, but here in the subterranean stem, the bud forming aerial shoot upward and adventitious roots downward to the soil. Also, the subterranean stem has scaly leaves covering the growing point or bud. Similar to the aerial stem in the point which has normal leaves. The main difference may be between the subterranean and the aerial stem is the absence of chlorophyll. It is not green such as the aerial stem, and only bearing scaly leaves to protect the growing point, to allow the stem to go forward inside the harsh conditions in the soil. There are several types for the subterranean stems, including rhizomes, tubers, culms, and bulbs. You will start with the rhizome. This is the typical subterranean stem, which has nodes and internodes, terminal buds, and aerial shoots, as well as adventitious roots downward. It can grow in any direction in the soil, maybe vertical, horizontal, or oblique way. There are two modes of branching for the rhizomes, either sympodial or called as cymose pattern. In this, each branch gives rise to the new axis. As you can see here, the old rhizome will be, or the branch will be at the bottom, and when we go, it grow more. The new rhizome or new branch may be at the top. In this way, the branches constitute a new rhizome. The other type of branching is monopodial or racemose pattern. They have single axes which give rise to lateral branches, such as wood sorrel. Like this one, the old branch will be at the top, while the small branch will be at the bottom, and the only small branches constituting not form an entire new rhizome. There are also several subtypes for the rhizomes, named as sopal, stolon, and sucker. The sopal is a long, thin, tough rhizome and rapid in growth with long internodes, such as couch grass. Stolon and sucker are similar. What is the difference between them? Stolon grow near the soil surface or just below the soil, such what happened in valeriana and licorice rhizomes, while the sucker grow under the ground, arising from the aerial stem, as happen in menta, or the top of the root, as in rosa rhizome. The second type of subterranean stem is the stem tubers. A stem tuber is defined as a swollen underground stem or part of stem, usually at tip. It's usually full of food material. The known example of stem tubers is the potato. It differs from the root tuber or other types of subterranean stem. They have bearing, as we discussed in previously in the previous slides. Subterranean stems is characterized by the presence of buds, nodes, and internodes and scaly leaves. Here we don't see the place of nodes or internodes, but we see what is called eyes. This eye constitutes of the bud scar and the scar of a scaly leaf, both of them constituting what is called eye. When this part of subterranean stem is planted under the ground, the bud scar started to constitute the new plant germinating or forming what is called budding of potato. So a stem tuber is a swollen underground stem, swollen due to its full of reserve food material, which is carbohydrate or starch, and is characterized by bearing small scales and buds forming what is called eyes and the terminal bud on the free end of potato. The third type of subterranean stem is the corm. It's shortened, swollen, erect underground stem. It's also swollen, such as the tuber, because it's full of the reserve food material, usually starch. And here we can see obviously the place of nodes and internodes, and also the place of nodes is covered with brown scaly leaves. Usually the leaves present on the subterranean organ is scaly, because if it's brittle it will be broken. It has to be scaly to can avoid or withstand the harsh conditions under the soil. And of course it is not green because it's not subjected to the sunlight, so no need for the chlorophyll to make photosynthesis, for example. So usually the scaly leaves present in the underground organs or underground stem is not green. As we can see here, it's scaly brown leaves arising at the node, and we have large apical bud and small axillary ones. Also adventitious root is appearing, as you can see here in this photo. For example, about the corm is the colchicum corm, which from which we get the anti-gout medication colchicine. It's very important. The last type of the subterranean stems is the bulb. Bulb is opposite in its definition to the tuber and cone. Bulb can be defined as reduced to small flattened disc and covered with crowded fleshy scaly leaves. What does definition mean? You know, onion and garlic that we eat at home. This hard part with fibers or hairs that we cut with a knife and withdraw before eating or using garlic and onion in our cooking. This part actually is the subterranean stem. This compressed hard disc is a subterranean stem, and these fibers or hairs arising from it is the adventitious root. So what we are eating, this is actually the scaly leaves present in the underground stem. So bulbs can be defined as small flattened disc covered with crowded fleshy scaly leaves. We have two types of bulbs named as tunicate bulbs and the second type is scaly or imbricated bulb. The tunicated bulb, for example, as we can see here, is the common onion, or other example is the squash. The outer scaly leaves are large and completely encase the inner bulb. Each scaly leaf including the inner smaller one, and they constituting like a circles around each other. This is the tunicated bulb, and we can see here the hard part inside the onion is the bud, and the hard disk down here is the reduced or flattened subterranean stem, and these are the adventitious roots. The scaly or tunicated, or imbricated bulbs, here each fleshy scaly leaf overlap at their margin for constituting what is called garlic cloves. The only outer scale, which including the other inner cloves, for example, that what happened in garlic, and also the inner hard part constituting the bud. This part is the reduced stem, and each clove of the garlic we use is entire fleshy leaf overlap at their margin and closed to form this clove. This was all about a brief hint about the morphology and types of subterranean stems. Now we will discuss the histology of subterranean stem. If we take a transverse cut in a subterranean stem, what are the tissues or cells we can see in this section? There are several tissues that we will discuss in detail now. It's resemble also in the structure, in their structure, the aerial stem, and they are devoid or containing very little sclerenchyma tissue. We have the epidermis, the cortex layer, the vascular bundle composed of precycle, phloem, and xylem, and inside we have the pith, like the aerial stem. At the first, the tegumentary tissues or outer covering is either constituting of the epidermis, as in couch grass, colchicum, and podophyllum. When the subterranean stem is young or newly developed or grown in the soil, so then the outer cover or outer tegumentary tissue will be epidermis. But when it gets older, it can be cork cell or mesoderm, more thickened cells to can withstand the harsh conditions under the soil. The tegumentary tissue, as I said, maybe epidermis, maybe cork cells, with or without rhytodoma and licorice, and also maybe what is known as mesoderm. The mesoderm is irregularly arranged superized cells derived from the outer row of the cortex as in white and black hellebore. What is different here is the thickening in the epidermis. Epidermis is usually parenchyma with thin wall of cellulose, but when this cells have been thickened by a substance called superine, so the cells become superized and the cell wall become thick here, not thin. This is called metaderm, and the cork is the highly thickened of all these types. This is what epidermis for young root, and when it gets older it converted to cork cell. This is because the presence of active meristematic tissue between the epidermis and the second layer, which is cortex or known as phalloderm. This active meristematic cells called the core cambium. It divided to form cork cells outside and phalloderm or cortex cells to the inside, so that the plant can grow in size and become older and bigger in size. The second layer in the subterranean stem is the cortex. Usually it's a parenchymatous tissue, acts as a storage area, as we told previously in subtypes of subterranean organs, such as especially tuber and corm, used as storage for the reserve food material such as starch. So usually we will find the starch granules baked in the parenchyma of the cortex layer. The innermost layer of the cortex, called endodermis, that could be distinguished or not according to the constituent in that layer of the plant. The third tissue present in the subterranean stems is the vascular bundle. Vascular bundle constituting of xylem and phloem surrounded by pericycle and transversed by medullary rays. Between xylem and phloem, there is active meristematic tissue allow the growth of vascular bundle in size, which is called vascular cambium. This tissue or this cambium is different from the corky cambium, but similar to it in its function. It's responsible for the growth of the vascular bundle, constituting phloem to outside and xylem to inside or inner world. And the vascular bundle is very important for the function of the subterranean organs. The xylem is responsible, for example, for transporting water, absorbed water and nutrient from the root to other plants. Finally, in the center of the subterranean stem, there are wide or narrow according to the plant, but there are parenchymatous cells called pith. Usually it's large area for storage and transport. It's similar to the real stem, and this layer is the major difference in the structure between the subterranean stem and subterranean root, as we'll see later on in the root. There is no pith in the center, but instead there is a solid mass of metaxylem, and so when we just look at the transverse section under the microscope of the underground organ, if we saw pith in the center, we can conclude it's a subterranean stem, while if we saw a solid mass of xylem in the center, we can conclude it's a root, not subterranean stem. So this is a very important information need to distinguish in pharmacognosy between the subterranean stem and root. Moreover, in the subterranean stem, we have two types of plant, monocotyledon and dicotyledon. We can use the transverse section of subterranean stem to can distinguish if this plant is monocot and dicot. Especially if we can't get the seed to see if the seed of this plant have one cotyledon or two cotyledons. This is the easiest way. But another way to identify the type of plant as monocot or dicot is to looking for the TS section of its subterranean stem. There are big important differences between monocot and dicot subterranean stem transverse section, as we can see in this table. At the first, in the monocot, there is no cambium occur between xylem and the phloem, and accordingly, this vascular bundle named as closed vascular bundle, which means it didn't grow in size. So the plant forming numerous vascular bundles, and we will see here it is scattered in the whole transverse section, not only in the stele region but also in the cortex region. They are numerous, but they are small in size due to the absence of vascular cambium. While in the dicotyledon there is a vascular cambium happened or occurring between the xylem and the phloem. So the vascular bundle here is named as open vascular bundle, which can grow in size. It's big in size, and we can see here the vascular cambium is distinct between the phloem and xylem. They are large in size, but they are few in number, and they are concentrated only in the stele region, not found in the cortex as in monocot. So this is a big difference in the shape between the transverse section of subterranean stem from monocot and dicot. The other point is endodermis. Usually in monocot it is not distinct, but in some plants it will be distinct, as we will see in in ginger transverse section. And in dicotyledon, endodermis is usually distinct. Secondary thickening, as we previously said here, there is no vascular cambium, and so on the secondary thickening not happened and no annual rings, while in dicotyledon, due to the presence of vascular cambium, secondary thickening can occur and annual ring is usually present. So it's obviously from the photo of the TS diagram we can conclude if this plant is monocot or dicot. So take a while to see this two real TS diagram of two subterranean stems. We can conclude that this one is a monocot because the vascular bundle are closed, small in size, numerous, and distributed in the whole TS diagram, while this one of TS diagram of dicot plant or dicot subterranean stem because the vascular bundles are big in size, few in number, and concentrated or present in the steel region only. The second part of this lecture, I will talk about the subterranean roots and what are the great differences between subterranean roots and previously described as subterranean stems. Subterranean roots can be defined as a part of plant axis that grow downward in the soil away from the light, and they are characterized by these are the differences actually between the subterranean root and subterranean stem. Roots are not divided to nodes and internodes such as the subterranean stem, and accordingly they don't bear any scaly leaves or any buds. It's also contain no chlorophyll, okay? And here the growing point is covered by a special tissue known as root cap or calyptra. In contrast to the scaly leaves that was protecting the growing point in the subterranean stem, we don't have here a scaly leaf, but we have a special tissue called calyptra or root cap. The root is constructed of 4 zones, as we can see here in this photo. The first zone, called the growing point, which is covered by the root cap, it contains the active meristematic tissue, allow for the growth and penetration through the soil. The second zone is zone of elongation, responsible for increasing the length of the root. The third zone of absorption, or called as root hair, constituting of epidermal protrusions known as root hair, which we will discuss in the next slide in the structure. But here what we will say in this part is it's responsible, this root hair are responsible for absorbing water and nutrient from the soil. They are non-glandular unicellular hair or prolongation from the epidermis. The last zone is the lateral branch of the root, which occupy the majority of root. It didn't appear here because it's another branch of the root constituting of the same parts. We have 5 main types of roots known as primary root or tap root, secondary root or lateral root, adventitious root, tuberous root, and aerial root. At the first, the primary or tap root is the first organ to appear when a seed germinates in dicot. For example, the radicle develop into a tap root. As you can see, when we plant the seed inside the soil, the radicle comes out from it, forming what is known as tap root. Such happen, for example, in Senegal and Belladonna and most of the cultivated plant when we propagate this plant using the seeds. The secondary roots or lateral roots are usually the branches arising from the primary or tap root. Adventitious roots are the roots that usually arises not from the seed or cotyledons as primary and secondary root, but from the subterranean stem. So if I will propagate the plant through seeds, This will give primary and secondary roots. But if I will germinate plant through subterranean stem for asexual propagation, the subterranean stem give rise to smaller roots known as adventitious roots. The storage roots or tuberous roots are roots swollen due to the presence of reserved food material. If the primary root is used as a storage organ, so it will be called tuberous primary root, and so on. If the lateral root is swollen, it will be called tuberous lateral root. And also the adventitious root, if it is swollen due to the presence of starch, for example, it will be called tuberous adventitious roots, and so on. For example of tuberous primary root, we use two common plants we use every day in our kitchen. They are typically tuberous primary roots, such as carrot and radish. Usually, as you know, carrot is root. Its type or is tuberous primary root. It's swollen, not very thin, due to the presence of nutrient inside it, such as starch and others, and also the same as radish. And we can see here, this is the primary tuberous root. It's swollen, and the other smaller one is the lateral or secondary roots. It's not swollen. Tuberous lateral roots, such as the sweet potato, you can see here the primary root is very thin, while the secondary or lateral roots are swollen due to the presence of starch. Aerial roots are some roots are hanging in the air to can afford certain environmental conditions the plant is growing on, or afford some functions. Usually they are adventitious roots, either hanging in air, such as in vanilla, growing down to anchor the ground, such as ficus, as you can see here in this fig tree. It's here to more support for the plant. Or they can be climbing and then called root tendrils, such as a hydroplant, or for absorbing water from air, such as in orchids. Now, this is the last part of this lecture. I will talk to you about the histology of root. There are big similarity between this part and the histology of subterranean stem, but actually there are very important differences from which we can characterize if this transverse section we are looking for is a subterranean stem or subterranean root. As we can see here, also the same layers except the base. We have here epidermis or cork in the cortex cell, and its innermost layer is endodermis. And finally, we have the stele region composed of xylem, phloem, and this is a very important feature. We have inside the center of the transverse section a solid mass of xylem in root, while in subterranean stem we have a pith. Epidermis, outer layer of cork of the cells or skin called epidermis. This is, as we discussed before, occurred in young root, but in old root the epidermis is replaced by cork cells due to thickening, due to also the presence of cork cambium, which can be divided, forming cork outside and cortex to inner wall. The root hair, as we see in the absorption zone of the root, it's an extension of a specialized root epidermal cells. So the epidermal cell could be protruded to increase its surface area, and so can absorb more water and nutrient from the soil, forming what is similar to the non-glandular unicellular hair known as root hair. This is to increase the surface area and absorb more water and minerals. The third layer in the histology of root is the cortex, and usually it's also parenchymatous and full with the reserved food material. Usually in the root we will see the starch or other nutrients stored in the parenchyma of cortex layer. The innermost layer of the cortex, known as endoderms. Then we have the stele region composed of the vascular bundles, which consist of xylem phloem surrounded by the pericyclic layer. Pericycle may be parenchyma or scleroids or fibers, whatever. Xylem vessels. We have here two types of xylem. We have living outer xylem, which is called protoxylem. This is the new formed xylem. And we have the old dead xylem vessels, which sent to the center of the TS, known as metaxylem. Usually metaxylem is responsible for the support, and protoxylem is responsible for transporting water. To give more explanation about this part, as we discussed before, we have what is called vascular cambium. Vascular cambium is responsible to form new xylem and new phloem tissues. So here vascular cambium in this layer forming new xylem called protoxylem. The new protoxylem is lignified and has a wide lumen, so it can transport water and nutrient from the soil to other parts of the plant. While the old metaxylem is highly thickened and usually it's blocked by the lignin cell, so we call it as a dead tissue. It can't transport water, but its main function is to support or anchor the plant or root inside the soil. The other part of the vascular bundle is the phloem or inner part. It carrying the dissolved sugars and organic compounds through the plant from the leaves to the roots. And finally, this is brief comparison between the differences between subterranean stem and subterranean root. At the first, we can see that nodes and internodes are present here in subterranean stem while it's absent in roots. Also, the scaly leaves present in subterranean stem and absent in root, as well as the buds, axillary buds, present here and absent in root. The lateral branches can be present in both of them. Adventitious roots present only in subterranean stem while it's absent in subterranean roots. Growing point here in the subterranean stem covered by scaly leaves and forming a bud, while in subterranean root the growing point is covered by what is called root cap or calyptra. The center of TS here in the subterranean stem constituted of parenchyma cells forming what is called base like the aerial stem, while in root it is formed of solid mass of metaxylem. So this is our lecture today. Thank you for hearing my lecture, and please, if you have any question, write it down below this video. Thank you.